Hi, hello, namaste. Welcome to Shrav's Biotech for You. Today we'll discuss about the second principle of biotechnology, that is bioprocess engineering or biochemical engineering. Bioprocess engineering utilizes living cells or their segments to get the desired products like enzymes, organic acids, antibiotics, vaccines, proteins, etc. Bioprocess engineering is divided into three major steps: upstream processing, fermentation, and downstream processing. Bioprocess engineering starts with the development of biological system, continues with the product isolation and purification, and finishes when the product is placed in a stable form. Coming to the upstream processing, today I am giving an example of bioprocess engineering to get a new rDNA derived product. The upstream processing starts with the development of biological system. It is usually an organism with desired protein or enzyme in case of protein modification. Upstream processing starts when an animal tissue is subjected to biochemicals in order to give us a desired gene. A plasmid DNA is then isolated from E. coli. These two are ligated to give an rDNA molecule. This rDNA molecule is then introduced into a microbial strain. Selection of microbial strain is characterized by the ability of this strain to synthesize the desired product having the commercial value. This strain is subjected to improvement protocols to maximize the ability of the strain to synthesize economical amounts of the product. After the insertion, it is subjected to plasmid multiplication and gene expression in order to result in a small scale culture. Hence, upstream processing involves preparation of the liquid medium, separation of the particulate and inhibitory chemicals from the medium, sterilization, air purification, cell culture, and harvest. Second step is fermentation. A fermenter or a bioreactor is a fermentation vessel used for the production of chemicals or biological reactions. It is a closed container with an arrangement for agitation, aeration, pH or temperature, and a drain or overflow vent to remove the waste biomass. It provides an environment for the microorganisms to obtain optimal growth and produce metabolites for the bioconversion of substrates into desirable products. Techniques for the large scale production of products are divided into two: surface or immersion, submersion or submerged techniques. In surface techniques, microorganisms are cultivated on the surface of the solid or liquid substrate, which is more complicated and rare. In submerged techniques, the microorganisms are inoculated in the liquid medium after the introduction into the fermenter, and they are stirred for homogeneous distribution of cells. In the fermenter, there is a large scale production of this inoculum or the culture. Moving on to the downstream processing. Downstream processing involves separation of the cells from the fermentation broth, recovery, purification, and the concentration to give us the desired new rDNA derived product. A vast array of methods like centrifugation, filtration, chromatography are used in order to obtain the desired product. The methods vary according to the chemical, physical nature, as well as the desired grade of the final product. With this, I wrap up today's session regarding the bioprocess engineering. Please like, share, and subscribe to my channel.